pointers here. You want to keep your focus on the wheel because you just never know. You can hurt yourself and you could obviously hurt someone else. So the university part under Myers, I'll send it back to you. Okay, thank you. Next, you want your first one.
Good morning, y'all. Hope you're having a good Monday. No, Monday. Yeah, today is Monday. I forget the days of the week sometimes. Hope you all had a good weekend. Welcome all of those who are here today. Welcome newcomers. Welcome oldcomers. Welcome those who are on the rebroadcast, the not so live stream. Just got back from Cleveland, Ohio this weekend, where I spoke at the Ohio Cichlid Association meeting. Like always, when I go to new places, I like to go to fish stores, and I also come home with new fish. So Preppers Kruger, hi Grandpa. Well, hello there. One of my kids is here today, watching me live. So I guess I'm going to get nervous. So what I want to do today is really just talk to you about some of the work that we are doing in Peru. Going to I'm sorry, I'm reading some of the comments. Liquid Zoo actually is pretty fun going there, just so you know. Just talking about some of the work we're doing in Peru on invasive species and things that invasive species have done all around the world. So, this is important for us as aquarium conservationists to know what to do with your fish once you're through it. Fish get too big for tanks. We have the Ohio Fish Rescue that takes them in. We have Florida Fish Rescue. And there are a few people here on the West Coast that will take in your fish if they are too big for your tank. So one of the things that we have to realize is when we go buy fish, is to buy fish that are appropriate. Fish are a long-term investment, just like your dogs and cats. And we have to be able to have the right aquarium, the right environment for these fish. And so we like to properly maintain these fish. We know that in 5-6 years, those fish are going to be much different size. So that small, cute little cuddly red-tailed catfish, maybe 4 or 5 years, is going to be a 3-4 foot long red-tailed catfish. Most of us cannot maintain those fish. So what do you do with them? The worst thing you do with them is take them out to your local stream and lake. Especially if you're in a cold environment, you're going to kill them. If you're in a warm environment, Florida, here in Southern California, there is a probability that they can make it in those environments and wreak havoc on what's going on. So with that, what I want to do is I want to share in my own share slides live stream. Okay, there we go. That's all good. My voice is very muffled. Maybe because I'm mumbling. Sorry about that. Give it a thumbs up if my voice is better now. Wait a few seconds to make sure it's good. No, you're not losing your hearing. If it's do anything that all is well, I guess that's good. So, much of my work in Peru, besides the research center, is that where I work on invasive species. Invasive species can cause dramatic effects on the environment. So everybody's saying no, I'm not sure what that no means. No, my voice is not good, or no, my voice is not good. How about now? Is that, I think that's much better probably. So can you hear Alishon, Jennifer, Freaky? Am I doing okay now? Perfect. Okay. For some reason, my microphone has switched on StreamYard. So all is jumbled. Well, we can forget about all that stuff to begin with. So let's talk about invasive species. Let's see if I can get this to work. Slide 2. So what we're going to look at is really the impacts of exotic species in the Peruvian Amazon. Good morning, Ian. So, exotics can have a lot of impacts to native fauna and flora. One of the first things we often see is they are predatory fish. Even if they're in their native environments, like some of the tilapia species that have been released all over the world, especially here in Southern California, in many cases, these fish were herbivorous in their native environments. But when they come to a new environment, all those niches are now open to them. They eat whatever they want to. And some of the tilapias that we have here in California switch from being herbivorous to carnivorous. And what they did is they went in and wiped out many of the native fish species that we had here in Southern California. We don't have that many native fish species, but like the desert pupfish in the Salton Sea area were wiped out by one of the things that wiped them out were the tilapias that actually found their way into those environments. They can hybridize with native species, and this, of course, means that the species have to be closely related. When I was doing some surveys in Cambodia, we see this with the native walking catfish, the Claris catfish that they have native to Cambodia. Claris catfish have been used in Africa for aquaculture, and they brought them to Cambodia for aquaculture purposes as well. And those that escaped into the native environments were hybridizing with the native Claris catfish. And what happens in many cases, you get hybrid vigor. Those species tend to grow, those hybrids tend to grow much bigger, much faster. They're more robust, they're more active. And what they do is they then outcompete either one of the mother-father species. And so you no longer have the original exotic, but what you've created an even greater threat with these hybrids. They can also modify the habitat, and in modifying the habitat, they can then allow other invasives and other invaders to come into the area. And lastly, they can spread new diseases as well as parasites to native organisms. Probably the most dramatic of the invasions by invasive species was that what we saw in Lake Victoria. Lake Victoria originally had more than 400 species of cichlids, and over 200 species were endemic to Lake Victoria alone. They brought in Nile perch to supplement aquaculture to the peoples of this area. They thought it'd be a good economic source for them. Nile tilapia, Nile perch can grow up to over 6 feet long, over 500 pounds, and they were brought in for cage culture. In other words, they put all these small Nile perch into cages into the lake. And of course, what happened as the fish grew larger, they broke through the cages, and they then decimated the native populations in Lake Victoria. And so what we have now in Lake Victoria are only 10 species of cichlids in the lake. And so you can see that they wiped out pretty much everything in that lake. Now, there has been some recovery. there have been some hybridization that occurring that will lead to speciation in the long run. But right now, Lake Victoria is not what it used to be at all. And that's the major impacts that some of these invasive species can have in their new environments. Now, Peru began their foray into exotics back in the 30s. At this time, the government introduced lake trout into Lake Titicaca in the southern part of Peru, and they brought it in to have a new food fish for the local fishermen. Also thought maybe it would bring in more money to these local fishermen that they could sell these trout. So you can see Lake Titicaca here. But what it did is it wiped out the native fishes in that lake. And these two fishes here of the Orestius genus were the two most important food fish for these fishermen. One fish here, Cuvieri, is totally extinct now. Lake trout decimated them, ate them till there was nothing left. And this here fish is now pretty well rare in the environment. So this just shows a native catch that they would catch. Since that point, though, Peru has brought in a lot of exotics. Most of these exotics were brought in because of aquaculture, others because of, just like in America, we have escapees from aquarium. So bighead carp, the tilapians, were all brought in for aquaculture. Mosquito fish were brought in for mosquito control, just like we have them here in the U.S. Now, in Iquitos, the two main exotics we're working with are one, the blue gourami, some people call it the two-spot gourami, as well as guppies. Guppies were brought into the area for mosquito control. There is dengue fever as well as malaria in the area, and they thought, ooh, this is a great fish to control these mosquito populations. My view is the local killifish were doing just as good a job controlling the mosquito population, but someone got into their brains and said, this is a bigger, better fish for the mosquitoes. Blue gourami were brought in the early 70s in aquaculture. There was a local fish farmer who brought in about 10, 15 species of exotic fishes to raise on his farm in order to sell within the local Peruvian aquarium market. What happened is that one season had exceptionally large floods and his ponds were flooded. All the fish were released into the river, and the only thing that really survived and multiplied were the gouramis. So I first started working on the two-spot gourami, blue gourami. So here's a close-up of a blue gourami. These are not inches, but centimeters. So don't think that this is a 10-inch fish, but those are centimeters. Now, gouramis have been a problem around the world. In Australia, they have a biosecurity law that says that any gourami brought into the country must have a quarantine period of at least 2 weeks. The reason being is that gourami have a iridovirus that can be transmitted to Murray cod. And Murray cod is one of the traditional fishers. Well, it's one of the traditional fish that the fishers fish for. It's very important to their culture. And in fact, Murray cod are considered endangered now. They've been outfished, they've been or overfished. They have been there's lost habitat due to habitat degradation. So they're trying to keep this fish alive, and through aquaculture practices, trying to reestablish it in the environment. So the Gouramis virus does not kill gourami itself. They are carriers, but they are known, this virus is known to kill Murray cod. So it's important not to bring that virus into the environment. So in Iquitos itself, here is a close-up of Iquitos, Peru. This is the Rio Amazona or Amazon River, the Nile River on the left-hand side, and the Itaya River on the right-hand side. The orange area represents where we find gourami in Iquitos. The origin of the gourami was in a fish farm in this area. Those fish farms are no longer there. They've now been bulldozed over, and houses sit in those areas. But so the original was in the Rio de Nile. But you can see that the distribution now extends across into the entire river. So it occurs in both rivers. Whether they actually disperse out into the Rio Amazon and this way, or somehow through canals that connect those two, no one really knows. For a long time, they wouldn't admit gouramis were even there. But we find gourami out in the middle of the rainforest now. Through practices, people are feeding them, collecting them and feeding gourami to Arapaima on some of these Arapaima farms that are out in the middle of the rainforest. So they're being dispersed naturally and artificially by humans as well. Now, during the dry season, what happens is the gourami collect and breed and multiply and congregate and have a good time in these open sewage canals. They occur here by the millions. Most fish in that area cannot live in these open sewage canals, primarily because of the lack of oxygen. But we know that gourami, with their labyrinth organ, can come to the surface to breathe air. And so they live just as well in this environment than they do in the open water environments. And you can see a gourami here. This little blue speck here is actually one of the blue gourami in one of these open sewage canals. You can see there's a lot of man-made stuff here. It's a collecting pile of refuge, of all manners of things that people throw away. Now, our collecting sites for these areas are in some of these open plains area, in the swampy areas. You can see how high the water will rise during the rainy season. So this is the low season. We go and collect fish, or we have people collect fish for them. I've come back with a few infirmities in my time when I was working in Cambodia. My tender white body just does not like being in the murky swamps, especially these swamps here that are part of the open sewage canals. I don't mind getting in the water, but these I mind getting into. So we collect fish here, bring them back to the lab, and do whatever we have to do with them. So what we've been doing is we've been working on both looking at reproduction and competition for food with the native fishes in the area. So let's first talk about reproduction. We know that the blue gourami, or two-spot gourami, Paul, are bubble nesters. Males will build a bubble nest, attract females into the bubble nest. They will spawn. The female will leave, and the male will tend the nest and tend the young until they're too many and too numerous and spread out too far apart for him to take care of them anymore. Now, what we see in many tropical regions, or regions that have a flood stage followed by a dry water stage is that reproduction tends to occur around the flood stage. So water floods into the surrounding forest. Those leaves and that forest material begins to break down. That material creates lots of bacteria, which feeds the plankton. And so there's a lot of food for those baby fish. And even if you didn't know what the reproductive season was like for a fish, what you do is you do what's called a gonadosomatic index, GSI for short. And so what this is the gonad weight in a ratio with the body weight. So here, what it says is that down here, you pretty much have no gonads. Up here, you have 10 times or 9 times the amount of gonadal tissue as you did regularly. So it's an easy, quick methodology to use. All you have to do is weigh both the gonads and the body. Normally, you do this with females since it takes more energy for the females to produce their gonadal tissue. And so it just gives you more information with females than you do with males. So this is a typical curve plotted over a year for those fish who have a reproductive season. Now, you could have 2 reproductive seasons. And so what you would see is 2 very distinct curves here. In this case, we only have 1 reproductive season. For those who breed all year long, this is what we normally see, is some maybe peaks and valleys, but no dramatic increase like we see in a fish with a very specific reproductive season. So what we did is we captured 50 fish every 2 weeks for a whole year, for 52 weeks. And what we did, we dissected all the fish. Since there was no sexual dimorphism in gummies, maybe some size dimorphism, but when you catch them, you can't figure out what's a male and female. We caught 50 of them, separated them out, male and female, and those females that we caught, we excised the gonads and then calculated the GSI. And so this is typical ovarian tissue for females. Bright orange, and if we had a close-up, you can actually see the glandular eggs in that gonad. So there may be, 4,000 or 5,000 eggs in that gonad that are being released during that reproductive event. So we weighed the eggs, weighed the body, and then we calculated GSI, an average GSI for all those females for each of those 2 weeks. And this is what we had. So we did get some peaks and valleys here, some low points, but these low points were caused by the fact that in this first or that last week of February, no females were caught. So all of the fish were males, but you can see that there are some peaks and valleys again, but no major peaks. So let's say there is reproductive seasonality here. What this says is that there's at least 4 reproductive seasons within that year. So 4 times more than our native fishes, which what means is that they can swamp the environment with young. They can produce more young than the native fish. Since there is some parental care, it gives the babies a little extra time. And so when they go out in the world on their own, so to speak, they're at somewhat of an advantage. So again, they're at a reproductive advantage to the native fishes here. We also looked at food. This is a internal view of the intestinal tract of a gourami. In their native habitats in Southeast Asia, they're known to eat zooplankton, macroinvertebrates, aquatic plants, terrestrial plants, detritus. So basically, they're omnivorous. They eat anything they can get their mouths on. And this puts them at an advantage in their native habitats because they're not specialists. They're generalists on what they're feeding. So again, what we did is we captured fish and dissected them, examined their gut, stomach contents, and then categorized what they ate by very specific categories. And so when you tease apart intestines of any fish, this is what you get. You get a big amount of mush. And through the microscope, then you can identify what they're actually eating. And so this is what we found. This took place in an environment where we had all fishes. In other words, this sampling didn't occur in these open sewage canals, but in a more pristine environment. And you can see the total number of individuals that we caught in this period. So almost 1,000 Guppies, Dyneema, which is Porthole Catfish, Astyanax, one of the tetras, Pimiolids, Pictus catfish, one of the small cichlids, Cyclosoma amazonorum, Triparthus angulatus, which they call the sardina. Some people here in the U.S. call it the giant hatchet fish. That's, not really correct. But, and so you can see detritus, plant fibers, insect larvae, Chironomus larvae, tubifex worms, fly or fly parts, algae, fish parts. You can actually identify ants. Some had fish. You can see the more carnivorous fish had fish, and others. Others included a lot of things that came from human in the environment. They included pieces of charcoal, rice, fish scales, a lot of things that aren't normal in their environment, but since people use this water as their to wash their dishes, all that stuff is going into the water, and the gourami and other fish will consume that just like it's a natural food. So we have all this data here. So, wow, that's a lot of data. What do you do with it? What we do is we compare the diet of the blue gourami to the diet of everybody else to see if there is overlap. And so what we calculate is called the Sorensen Index. It looks at the amount of a particular food item, the amount of different food items, and we do the comparison between the two. If we have a Sorensen Index above 60, that indicates that these fish are in competition with each other. And you can see here, wow, big old blank screen. I hope you can, hope there's not a blank screen there with everybody, but for me, there's a big black screen in the middle. And what it says is that here we have these fish here that are in direct competition with the gouramis, the cichlid, the sardina, pictus catfish, porthole catfish, Hemigrammus pulcher, the pulcher tetra, the banjo cat, the dwarf wolffish, as well as the red tail or pink tail calculus. Now, Crinocycla, which is the pike cichlid, 55%. So there's almost an overlap of diet with them. If you collect more fish of Crinocycla, we caught 5 or 6, that may boost up those numbers. So you can see that they are in direct competition with the native fishes in the area. And since they occur in higher numbers, they are probably outcompeting the native fishes in those areas for food. Now, another thing we looked at is thermal limits. As global warming increases around the world, it's going to cause dramatic impacts in freshwater environments. Most of the time, we look at what happens in the ocean because of sea level rising, but there is an impact on freshwater environments. So what we did is we looked at the critical maximum temperature in in guppies, as well as the one of the native killifish, Rivulus rectocaudatus, which is the red-spotted killifish. In streams that we find guppies, we don't find rectocaudatus. In streams that we find rectocaudatus, guppies haven't invaded. So what it appears is that the guppies are out competing the rectocaudatus, and in fact, we have done some experiments here at the university that have shown that when guppies and gouramis are kept in the same tank, guppies tend to be more aggressive for food, and that in turn makes the killifish more aggressive for food. They spend more time out in the water column, which makes them more prone to predators. When rectocaudatus are found by themselves, they tend to stay under leaf litter. So it's not like the guppies are acting as dither fish, because in the native environments, that's where killifishes are normally found, under the leaf litter. You don't find them swimming out in the open. So they're just doing their normal behaviors. We also compared gourami to the 3 top competitors for food items in their environment: the sardina, Amazonorum, and the porthole catfish. So to do this, you have the fish in a small tank. You add warm water to them 1 degree at a time to see at which point that they basically do not move anymore. The fish aren't dead. As soon as they reach that critical max, we put them back into the normal temperature and they recover just fine. So none of these fish were killed in the act of these experiments. So let's see the critical temperatures or the thermal limits first for guppies here and rectocaudatus here. So you can see that the guppy critical temperature is about 100 degrees, whereas the critical temperature for rectocaudatus is down into the 90s, low 90s. So as temperatures increase, the rectocaudatus would die off, leaving the environment solely for the guppies. If we look at what happens in the gourami and their competitors, here is the gourami max temperature, again, above the hundreds. Amazonorum is about 37 degrees, is body temperature 98.6. And the 2 other fish, the porthole and now I forgot what fish is. The other fish, you can see that their thermal max is much below that. So as temperatures increase, gets a point above the cichlid, you will have then the you will have the gourami. And even if it doesn't get as high to kill off the cichlid, you'll have an environment that is based totally upon the gourami and the cichlid itself. So we can see that we will have problems as temperatures increase. Now, one thing that we just recently found is there are now tilapia in the environments. A few years ago, we did some environmental DNA work where you collect water samples and then identify what fish are found in the water samples by looking at the DNA in that water sample. At that point, we did find some tilapia DNA. We did not actually capture any tilapia. A couple years ago, we actually found tilapia in some of these streams. This past summer, we found even more tilapia. They had an extreme flood last year, and tilapia, we know, escaped at least one facility that was using tilapia to feed their Arapaima as well as their crocodiles, their caiman on this one farm. This farm flooded. We have tilapia in some of our ponds now because we're right adjacent to that Arapaima farm. So we have them in our ponds. We're actually removing them as quick as we can, but tilapia reproduce very rapidly. Fortunately for us, they're not in our experimental ponds. They're in a couple ponds that are catch-all ponds for us. So it hasn't impacted the research center per se, but it has starting to impact the native environment. So unfortunately, if tilapia get established in the rivers and the streams, you may have a fish-free environment with tilapia, especially some of the smaller fish, the tetras, some of the smaller catfish. Tilapia will munch through everything. Now, that's the end of that. So I just want to let you know that most of you know you can follow us a lot of places. Go to our website, amazonresearchcenter.org. We do have a Facebook site. We are on Instagram, _Peru. And of course, you're all watching me here on YouTube. They are on we do have this YouTube channel that we have a couple things that we have. One is this weekly live stream. Another is we do a once a month Saturday stream through Zoom. And if you want to, get on the list to be invited to those Zoom events, you can go and email rmays at amazonresearchcenter.org. And what we do is we post them after we have the event, so you can go look at those people that we've had on the Zoom events. There will be 2 more posted coming up. Our next Zoom event is the 23rd of April at 10 a.m. Pacific time. And we'll probably have an individual from Australia come on and talk to us about aquarium conservation, some of the work they're doing with aquarium conservation. Now this summer, we're going to start again the internship volunteership program. So if you're interested in doing an intern or being a volunteer at the research center, if you go to our website and you click on Get Involved, you can apply for an internship or volunteer program. The only thing we ask of you is you spend at least 3 weeks there to conduct the internship or volunteership. All you have to do is find your way to Iquitos. Right now, plane tickets are going for about $800. You spend 3 weeks minimum there. We house you and feed you at the research center. So there you can either work in the public aquarium, help us do research, do some of the things that we're doing at the research center. And you all know that we just conducted our raffle. The raffle will be announced live next Monday on this live stream at 10:30 a.m. So we will have the drawing live and in person on that live stream next week. We were going to do it today, but there is a club that bought raffle tickets that are going to be raffling them off to their members. You know, it wouldn't be fair to that club if we already had the raffle today and they're raffling off a losing ticket. That probably would not be the best money making for them. So with that, I want to end this portion of the live stream and go back to me live. And we'll go through some of the questions that I saw that came up in chat. Again, if you have a question, please do it at amazonresearchcenter.org. Yes, Paul Sotero, I mentioned that. This is the blue gourami, Trichogaster trichopterus. And it was a fish that was brought in to raise and sell in Peru as an aquarium species. That's, I think, the only question I can see. I have top chat on. Sorry, not live chat. So Paul asks, is there any evidence of the larger native fish predating on the gouramis and tilapia? No, not of right now. We know that, again, both of these species are used in the aquaculture industry to feed the aquaculture predatory fish, the Arapaima, primarily. Then one of the problems with tilapia, or not tilapia, with gouramis is that they're catching the tilapia in these open sewage canals. Now, question could be asked then, these diseases, these parasites that the gourami are harboring, are they being transmitted to the Arapaima and then potentially transmitted to humans, since these Arapaima that are being raised are intended for human consumption? That's one thing we tried to look at, but what we're able to do because of the potential hazard is that we were able to get a local law passed, a local guideline for the aquaculture farmers that prevented them or prohibited them from using gourami as a live food for their Arapaima. But as we know, any law is as good as it's being enforced. And unfortunately, this law is not being enforced. So you still find people selling gouramis, and they sell them for about a penny a piece, maybe as little as $1, $2 for 1,000 gourami to the Arapaima farmers. And so, it doesn't do any good to have a law if it's not being enforced. There are caiman in this region of the Amazon, but in the 14 years that I have been in the waters, I have never seen a caiman. The only caiman I ever see are on that fish farm that has large caiman, or in the marketplace where you find caiman cut up for consumption, or they're actually grilling the caiman. Whether these are caiman that they've caught in the local waterways or they're caiman that come from the caiman farms, you really can't tell. They won't tell you where they're coming from. So caiman have been fished out in the area, and I've only seen one snake in all that time too. So if you're afraid of snakes, not going to happen. Not going to see them. Jennifer Weaver asks, what's the status of the newly discovered red pencil fish? No one knows. I mean, it was just recently discovered. I actually have talked to our resident manager there to try to see if he can find exactly where it was collected, to see we can then go in when I'm back there in June, go there and do some non-destructive sampling, do some environmental DNA work to see if we could identify any other populations there. Not so they can be exploited, but potentially so they can be protected. Since it is just a newly discovered fish, no one knows what the population status is. So that'd be a good thing to look at before this thing is starting to be exploited into the hobby, because it sure is a beautiful fish. All that red surely makes it a much more desirable pencil fish than the normal red pencil fish, which is critically endangered right now. So we hope to get on that to see what the status of the populations are. Any other questions you all have? Questions about anything? Questions about fish? I really don't know much about fish. I just kind of pretend I do. So I am Dr. Pretender. That's what you can call me. So if not, I guess we'll end our stream today. I hope you have a good Monday. Be kind to your fish. Do your water changes. And that's it. I hope everybody has a good day. Oh, Jennifer does ask, what species of pistos do you find in your area? See it, Jennifer? I mean, people wanted to go away. Now you ask the question. We have cockatoides. We have Apistogramma bitiniata, Apistogramma agazzizes. There is, which are now very rare in the wild, Apistogramma altotepici, which is an undescribed species of Apistogramma that is undergoing description as we speak. These are this is an Apistogramma that gets to be, the males get to be about 3 to 4 inches long. When they're breeding, they're a beautiful purple blue coloration. Because of that, they were overfished. We have shown that they can successfully breed and really have good numbers if brought into aquaculture situations. You don't have to feed the fish. These fish feed off the natural insects, the cladocerans, the daphnia, the other microinvertebrates that are in the water. So it's a fish that we're going to use for demonstration purposes to teach the local fishermen how to undergo aquaculture for aquarium fishes, especially with some of these more rare species that bring good money. When you find altotepici on price lists for the exporters, they go for $25 a fish. So it gives them a good money, gives a good income for these local fishermen that may make during the rainy season where they can't fish. They probably bring in maybe $10 a day at the most. Prepper crewers learned that Arapima have milk that they can cause heightened parental care over their young. Have you ever observed this behavior? No, I've never observed this behavior. I have seen the corralling behavior of the Arapima, but I have never observed this behavior in live Arapima or dead Arapima, as the case may be. But thank you for that question. Yes, Paul, pistos tend to be good breeders in captivity, especially when brought into aquariums, but most of these fishermen cannot afford aquariums. And so we're trying to show them how they can breed these fish in ponds, which can be very, very easily dug by these local fishermen since the water table here is so high. The ponds fill up with water very rapidly, and then they can just throw in 3 or 4 pairs into that water, come back 3 or 4 months later and harvest the young. And they can harvest 100, 200 young if the pond is large enough. So it gives them a nice income It'll probably cause these fish, the Alto Tapichi, to come down in price, which may be better anyway for the hobbyists. They'll become more popular in the hobby, which then leads to more demand, which leads to more aquaculture ponds. So we think this is a good model. We're not telling these fishermen don't collect fish. That's in their livelihood. But what it means is they don't have to over collect during the dry season when that is the only time they can collect fish for income. The rainy season, it's too hard to collect fish. They can collect some, but not in high numbers like they do in the rainy season or the dry season. Okay, that's it. Thank you all for your questions. Thank you all for coming. Hit the like button if you like it. Hit the share button if you like to share. Please subscribe if you want to subscribe. If you don't come back. And one last thing for my friend Papa Shrimp. I like shrimp. So thank you all today. Have a good day. Be kind to each other. Be kind to your fish. And with that, I will end this broadcast. Goodbye.